I study epilepsy in my lab, and the vast majority of the epilepsy field is really focusing on either the children, rightly so, or adults, but not really truly the aging-related seizures.
The peak times in life where you're likely to have a seizure and have epilepsy are really early in life, and then much later in life as you're getting older. We know there are a lot of kind of genetic and other causes early in life, but the later in life is really underexplored.
The Knight Initiative had a call out for collaborative interdisciplinary grants to advance solutions, novel solutions, out-of-the-box solutions to the problem of aging. And I started to read papers on aging for really the first time in my life. I was stunned when I read Katri Andreasson's paper, the molecule that she identified as playing a huge role in aging-related neuroinflammation and cognitive decline was a molecule called PGE2 that was actually the same exact molecule that we identified as playing a role in what happens after seizures in the normal adult brain.
And so if we know that seizure activity increases with aging, that could potentially be driving increased PGE2 production. We wanted to see if that was a critical link and then target PGE2 signaling and use an intervention to try to reverse that in aging and restore normal cognitive function. So we started to meet with Katri's lab and Song Wang, who is an instructor in Katri's lab.
And Katri Andreasson happens to be not just at Stanford, but also in my building, just one floor down. So the connection was born that way.
Having the grant from the Phil and Penny Knight Initiative for Brain Resilience allowed us to bring together our different approaches to science to really address these questions. They have a lot of great tools for looking at aging in animals, older animals, and looking at molecular pathways, neuroinflammation, how that's changing. And then our lab has great technology for actually looking into the brain at how neurons are talking to each other, and then the molecules they're releasing and how we can restore that normal function in aging.
So we're giving treatments to the animals. We're using young mice and old mice, and these treatments are basically so we can actually see the effects of blocking the PGE2 accumulation and the related inflammation that comes along with that. So far we have seen a result in the memory performance of aged mice that have been treated. It's as if you've had a hard time finding your car for a long time now, but then you get this treatment and all of a sudden, there's my car, and it's really exciting to see that maybe we've got something to maybe help people with their memory.
The collaborative grant that we received not just resulted in scientific advance that you can measure with papers and and actual experimental data, but it truly transformed a good part of my lab.
I'll be applying to grad school, and I want to continue on with this research because I want to see some progress related to brain resilience. And we have a long way to go, so seeing the dial move just a little bit is exciting. We got to keep pushing it.